and biofertilizers, $1.3 billion of the market. That's a lot of change. And as we're starting to look at, can we move away from chemicals and those type things, and can we start to introduce things like biopesticides to do those jobs, this is going to be important as we move forwards. So when we break out biostimulants and where they're used, you can see a lot of biostimulants currently are used in the foliar phase. I think this is done because it's easy. Most guys are putting something on through a sprayer, so it's as easy to add it into the tank. When we start thinking of some of the products out there that are added in, it doesn't matter whether it's a Radiate from Nutrien, whether it be a Bioforge from Stoller, whether it be our Transit S product. They all go through the sprayer. They're all very timely. Okay? The soil was starting to grow on that one. We're starting to understand that we need to influence soil biology. Okay? So we're now starting to concentrate and focus more. But we're moving away from the simple things like humics, and now we're moving in to actual biological products that contain bacteria and fungus. The seed's more challenging because, again, if you're not on the seed when the grower takes that seed, it's difficult to get on that seed. And a lot of that would probably account for inoculants. So when we start to look at the difference between a biostimulant and a nutrient, what we see is nutrition contributes to a lot. Improved plant health, chlorophyll, biomass, abiotic stress tolerance. But what we find is there's one thing that the nutrition side of things, even though we make that plant healthy and more proactive and able to shake off stresses, we find that biostimulants are going to help us reduce things like abiotic stress. Okay? So when we start to look at it, this is what some of the biostimulants, what happens when plants go under stress. Okay, there's a lot of different things up there. Small slide, very busy. So let's blow up the water deficiency. Very apt after last year. Okay, a lot of guys last year farmed under drought conditions. The crop suffered. We didn't know what to do. So we stopped. But when a crop goes drought deficient, what we see is water potential reduction, cell dehydration. We see wilting in the plant. Okay, those secondary effects then become reduced cellular metabolic activities. We see stomata closure. The plant tries to protect what water it's got. Okay, there's a lot of different things that are going on in the plant. How can we influence them positively? We're going to use biostimulants. So this chart here, this is a chart of all the different types of biostimulants that we can use. It's confusing, isn't it? And as a grower, I'm faced with that every day. Someone comes to me with one of these groups every day. Do I want an acid, a microbial, an extract, or one in the other? But we don't really know where they fit. So what I want to do is kind of make that complex simple. So what we've done is taken all of that noise on that last slide and put it into a simple wheel. So in the middle, you can see the word biostimulants, and around the outside are the 5 groups that all of those things fit into. So we have a microbial group, plant extracts, other, carbon sources, and amino acids. And we're going to run through these really quickly and kind of position some of the products so you have a better understanding. So we're going to start with the plant extracts. You guys probably use these a lot. It's probably one of the oldest groups that we have most knowledge on. And into that group fall things like the seaweeds, yucca, kelp, polyamines, auxins from seaweeds. Brazinosteroids is a new one. But we all understand the seaweed group. We use them quite a bit. Challenge is when we get to seaweeds, it's not that simple. There's multiple types of seaweed, and each one has its specific arrangement of hormones. So what you see over there is we've got Ascophyllum nodosum, which is a Kelpac product. And then we have Ecolonia maxima, which is an Arcadian product. These differ by their extraction methods. One method uses acid to break it down. One method uses a high-pressure extraction. So it gives us a different type of hormone spectrum. And what happens is it means that all seaweed products aren't the same. So if we take the seaweed product on the first side, you know, that could be a wine grape. It's going to be a quality. It's going to give us a bottle of wine. But then we start to look at that other type that's extracted slightly differently. Now we get an orange. And now we make orange juice. So we need to know what those products do and how they're going to affect the crop. Because if one's loaded with more cytokinin, that's going to influence rooting a lot differently to one that's not. And this just goes into that discussion. So we can see the E. maxima, the A. nodosum. When we start to look, the maxima affects more on the rooting side of the plant because, again, it's got more of the cytokinins and the auxins in. When we go over from the nurseurium, the above soil parts are affected. But generally, when we use both and add them together, we can see that the effects are pretty similar. But what do we want to do? If I want early season root growth, I'm going to use the Maxima product. I'm going to encourage those roots to go down. I'm going to trigger auxin. Cool, this is annoying. So when we start to look at it, there's 5 different plant hormones that are known at the moment. 5 classical hormones that we're all well aware of. Growth hormones, auxin, cytokinin, gibberellins, all influence what the plant does. I'm going to pick on auxin because it's one that we hear a lot about. Auxin triggers root and shoot differentiation. Basically tells the plant you've got enough green stuff, put more white stuff down. Do you think that would have been a benefit last year as we dried down? As we pushed those roots down to follow the moisture, follow the nutrients? And then when we start to look at some of the other hormones involved, we've got ethylene. Ethylene's a stress hormone. Should rise at the end of the season to tell the crop to die. But any time we get stress like high heat, waterlogging, drought, that ethylene will rise and it will cause that plant to go reproductive. So when we look at the terminology and how these companies approach the market, some companies will tell you, hey, our product's got nutrients, organic acids, alganates, auxins, steroids. Doesn't help you. Then we get other companies that talk about organic acids, alginic acid, amino acids, mannitol, etc. They start to break it out into the different groups. And we need to start to understand what those do. So auxins, what do? Why am I worried about auxins? We know a lot about auxins for rooting. We need it, root and shoot differentiation. But did you know you need auxin at the end of the season? Because auxin is what tells the pollen tube to go down and fertilize and make the seeds that you need to harvest. That's interesting because we need zinc to produce auxins as well. But also when we start to look at some of the interactions with auxins, we can see a lot of the other hormones are starting to interact. Okay, so what we get is auxins are going to help produce the root, but alongside that we're going to have cytokinins, acetic acid, polyamines, all contributing into that plant's growth process. So how do we know this? One thing that we do with any hormone to see how it works is we do a mung bean assay. Basically what you do is you grow a mung bean for 10 days, cut the roots off, dip it in the solution you want to test, and we're looking for how does that re-influence the rooting of that plant. So what we can see is our check there, smaller roots, not overly fantastic, but when we add the seaweed in, look at the different number of roots, look at the root benefit. And that's because we're triggering the auxin in the plant to tell that plant to make roots. Polyamines. Polyamines is something you'll hear about. They're good stress relievers. Again, they're going to improve rooting alongside the auxins, but also they're involved in that pollen tube and germination growth. This is the beauty of seaweed. So rather than trying to put these on separately, they're all in that one thing or product that you buy that contains the seaweed. So when we look at polyamines, what do? So we're going to do a drought stress recovery. So again, very poignant to last year. So this is what the plants look like. We transplant, dipped in foliar 14 days later. Now we leave them for 18 days without water. And then we water them one day. After watering, you can see that plant on the right-hand side all the time, which was treated. It's bouncing back quicker because we're triggering that stress response in the plant. We're helping that plant get over that ethylene shock. Last thing I want to move into on the seaweed is steroids. This is a new word that we're using. And why are we using it? Because analytics have got better. They've been able to find this and identify what it does. So what the steroids are important with, they're important for pollen tube elongation. So they fit with the auxins. Chart here shows that. We look at that top line there, the one in that orangey color, that's steroids. Actually produce a better pollination effect than the traditional auxins. Why have we not spoke about this before? Because we couldn't identify it. The strongest response that we could identify at the time was auxins. So now we're going to move into another source which is quite poignant at the moment. We're going to move into carbon sources. So we're going to move into the carbon sources because this is another buzzword out there. How many of you guys have heard of humic? Nearly everybody. Humic's a great fix-all. What about fulvic acid? Okay, great. Polyphenols, tannins? Any of those? Heard of those? Good. Because the thing is, those things are actually in those two first actives that we spoke about. So as we move on, what we've seen is a progression in the organic acid sector. So we started out with humics, okay? We put the humics in the soil, we fixed the soil. We didn't quite understand why, but what it is, it's feeding soil biology. And in a humic product, you do have a portion of fulvic. So the humic feeds the bacteria, the fulvic feeds the fungi in the soil, which is contributing to a good soil biology. The challenge is we're using big rates, and we've never been able to put enough or never really seen performance at the rates of the humic that we're using. So what we're trying to do is move towards refining. What do we want out of that product? Do I want the humates? Do I want the fulvics? Okay? So as we refine that product, what we do is we take a lot of the insoluble stuff that has no activity out. And that leaves behind the hydrocarbons that are more active and have more benefit. And basically what happens then is we've got to chemically characterize this so we know what it does. What genes in that plant is it turning on? Rather than me just coming to you and saying, hey, do you want to buy some fulvic? It makes your fungi grow. You know, what genes does it turn on? So when we go through this, the reasons we move away from humic still has a great fit, but look at it. Mainly amends the soil at a rate of 40 litres an acre, so there's only one place that fits, in with your fertilizer. When you put it down the spout. Okay? But what we see is big handling issues. There's a lot of bulk, a lot of product. So when we go to the fulvics, what we do is we take out those humates, we take out that a lot of the carbon source. It's better. Now we can get down to a rate of 1 litre an acre. But now we're only influencing the fungi in the soil, not the bacteria. Okay? Still a challenge, though, because if I want to take a fulvic and put it in a product with nutrition, that's a big rate. You guys are going to be hauling a lot of stuff. So what we did is we said, well, can we take any more of it out? So what we have is a product called CPPA, which is where we've taken out a lot more of the inerts, get it down to a low application rate. We've classified it and characterized it. We know what it does. And now we can actually start to co-formulate it with nutrition. And we'll get to the reason why I need my biostimulants with nutrition shortly. So basically, this is what it looks like. So when I start to look at a humate, what I have in here, I've got lipid-like substances. I've got humics. We can see them in the bottom left. I've got enriched in CPA. We've got tannins, CPPA. And then in our seaweeds, we get a lot of lignin-like products. So that's what our humate contains. Okay? So what of those do I actually need to get the benefits? Well, tannins are going to be polyphenols from plants. Lignins, they're the fibers from modified by fungi. Lipids and steroids, we can see them up there. They're the enzyme-driven responses, so they're going to be helping there. Condensed hydrocarbons, that's the stuff that's going to be derived from plant material. Like the hydrocarbons are things we don't necessarily need a lot of. So when we start to look, so what we've got at the top there is we've got our humics. Traditional lenodite, coal humic. You can see where the activity is. Every one of those dots is a point of activity, and we can see it falls quite nicely in amongst there. Okay? When we go down to the CPPA at the top, we can see that we've moved across. So you can see on this bottom one, the lenodite, it's a lot of it's in that humate side of things, okay? So we're not as active. When we compare it to a fulvic, and I mean a fulvic should be more active because we've taken a lot of the inert out and we've kept the active. So we can see we've shifted that across. We're more in those lipids. But when we start to look at the CPPA, which is highly refined, it's the tannins that we've kept. So when we start looking at peak responses, because this is important, how much do I need to put on to get a response? So humic acid, ideally to get a response, 100 to 300 ppm, which is that 40 litre an acre for 3 years to amend the soil. So there we go. Now we know how to do soil health. All right. If we use 4 litres an acre, which is more traditionally used, that's used to stop the tie-up of phosphate fertilizer. See, that's a great use. We move into the fulvics. Now we're moving more into a foliar application. 30 to 50 ppm we need for a peak response. 2 litres to 7.5 litres an acre, still a little bit challenging. Into the CPPAs, half to 1 ppm, 100 mils per acre foliar, 200 mils per acre soil. So it starts to fit easier with your programs. And when we talk about characterization, this is it. So what this chart actually shows here is what genes are turned on and what genes are turned off with the CPPA product. Okay? Left-hand side of the screen where you see a minus, that's down regulation or turning off. Right-hand side of the screen, turning on. So we're going to turn off things that create ethylene and ABA, the stress hormones. So now we can reduce stress in the crop. We're also going to turn off receptors that bind proteins, receptors and proteins that bind calcium, zinc, manganese, etc. So we're going to have more mobility in the plant of those nutrients. We're going to turn on things or upregulate transporters of calcium and zinc to help get them into the plant, move them in the plant. Okay? And then we're going to turn on a lot of the transmembrane proteins and also that energy production, that ATP cycle in the plant. Okay, we'll rock on. When we start to look at CPPA improving nutrient deficient delivery, we can see the numbers here. What we get a 12 to 22% increase in phosphorus, 20 to 50% in zinc and manganese. And again, consistently, when we add these organic acids in to a copper formation, you can see that we get a return in leaf concentration. It shows we're taking those nutrients into the plant. What we find, though, is when we add CPPA to the seaweed, so we get synergies. Seaweed and check on the left-hand side of the screen. When we add the seaweed and CPPA on those bottom plants, we can see because that product triggers auxin in the plant alongside the auxin in the seaweed, we get a better rooting. Under drought conditions, you can see again, control on the right, CPPA on its own, seaweed on its own, but that combination together, managing stress, triggering rooting, really does benefit the crop. Right, we're going to run through amino acids really quickly. Amino acids, these are going to be peptides, enzymes, metabolites. Whoops. So what is a metabolite? Metabolites include enzymes, organic acids. Okay, but basically what they are they're molecules that are produced by bugs that we ferment. So when we use the bugs in the soil, it's actually some of the metabolites that we're using. So how do we get there? We make bugs, we ferment them, we filter out the bugs, we use the metabolite. That's as simple as it is. That's what a metabolite is. Okay, why are metabolites better than live bugs? Shelf life, usually. Okay, they last longer. And when we start to look at some of the metabolites that we have, low molecular weight organic acids going to give better phosphorus solubility. That's got to be a good thing. Amino acids, again, phosphorus solubility, mobility of inorganic nutrients. We've got peptides that are going to stimulate soil microbial metabolism. And we've got non-protein amino acids in there that are going to help with damage and resistance. Amino acids. So basically what do? Chelators are nutrients, but also they can influence things like, just looking at the slide, opening and closing the stomata, stress relief. So metabolites are optimizing the product that we look at, M2. We filter out the bugs. We're looking for the phenolic acids that are going to give stress responses. We want flavonoids to modify auxin in the plant, get better rooting. Okay, and we want the polyamines because we need to manage stress, because we always said crops always under stress. What you can see here is when we start using these metabolites, the soil on the roots on the right isn't just stuck there because. What we've got is a healthy plant exuding sugars and helping feed that soil microbiome, helping that soil to stick to the roots. And again, when we start to look at this is in the Pacific Northwest, we're looking at yield increases over and above where we add in the metabolite products. Also, when we start looking at amino acids at flowering, what we start to see is that when we use things like glutamic acid, proline, etc., we get a nice benefit again because we're starting to trigger auxin responses, we're helping that plant pollinate better, and we're managing pollen hydration. So, why are we using a metabolite? Well, we want to use it to increase that rooting of vegetative plant growth. We're reducing impacts of abiotic stresses. We want to improve nutrient efficiency. You know, sorry guys, I mean, we're all under fertilizing. Our checkbook doesn't allow us to get to where we need to be. So let's make sure we're using that fertilizer as efficiently as we can. And it also improves that crop performance. Last topic we're going to run through very quickly is the microbials, Rhizobium, Bacillus, Trichoderma, Penicillin. You've heard them all. Lots of bugs in a jug. What are they? Bacteria and fungi, because we need both in the soils. We're hammering our fungi content in the soil because we use a lot of fungicides. So basically where they are, though, they're all around. They're on the leaf, in the leaf, and around the root rhizosphere. So we need to manage this. Okay. Tell you what we'll do. We'll whiz through this last bit. Because I want to get to the can biostimulants replace nutrition. Okay? Can biostimulants replace nutrition? Fuck me. Or do they complement nutrition? So, check on the left-hand side, nutrition in the middle. Nutrition works. We all know that because we do it, don't we? Okay? In the middle, we can see biostimulants. They have an effect similar to nutrition. So maybe they could replace nutrition. So I can choose where I spend my $5. However, when I look on this right-hand side, look at the difference in the leaf, stem, flowering when we add the two together. And what happens if we're not careful is we have nutrition on its own and a biostimulant on its own in the middle, and we're actually telling that crop to go nutrient deficient. Because we're telling it, don't get stressed, everything's okay, make more roots, find more zinc, but there's no zinc around the roots, so the crop goes deficient. So if we don't add a nutrient to that pack, the crop's going to go deficient. Last slide. So in summary, we need to understand what they are or use a credible supplier that can tell you what they are and how they work. Understand how they work, important. Understand when to use a particular technology, because they all have different timings. Need to understand the plant. Plant's always under stress, that's a given. And there's a synergy between biostimulants and nutrition. Unless we're over-fertilizing, we'll always need a nutrition pack to drive that response from the biostimulant. And again, biostimulants do complement nutrition. Thank you.
Thank you. Thank you. Is this working? Yes. Thank you. And sorry to put you under such pressure
That's
here,
fine.
but we have to stay on time here with the program. We have 6 presentations this morning, and actually you're the next one also.
Yep.
So
This one will be quicker.
take a minute for you to catch your breath here.
Grab some water.
So sorry, I didn't have your actual biography here to explain, to introduce yourself. I introduced Jarrett Chambers before. But anyway, welcome everyone here. We'll continue with the program this morning. And we're on field innovation and knowledge. And we're talking about this morning, the first presentation was clearing the confusion on biostimulants. So I'm sure everything is clear straight in your mind now after this presentation. There is an exam on the way out that you can pick up and fill and see how you pass. But anyway, no, just kidding.